Good afternoon, dear students. Today we are going to discuss about transduction. Last time we discussed about transduction, and that to the first type of transduction, that is generalized transduction. Today we are going to discuss about specialized transduction. So first of all, let us discuss, all of you know by now what recombination is. What is recombination? Breaking and rejoining of two parental DNA molecules to produce a new DNA molecule. Okay, what is transduction? DNA, in case of transduction, it is a type of genetic recombination in which DNA is transferred from one cell to another cell with the help of viruses. Or you can say that bacteriophages are the vectors that carry DNA from donor cell to the recipient cell. There are two types of transductions: generalized transduction and specialized or restricted transduction. In the last class, we discussed regarding generalized transduction. What is generalized transduction? It was discovered by Zinder and Lederberg in Salmonella typhimurium, where they found that there is a bacteriophage, that is P22 phage, which transfers DNA from one bacteria to another. But any part of the donor DNA, any gene of the donor DNA in general is transferred from donor to recipient. So the name generalized transduction. So, okay. So any part of the bacterial genome from one cell is transferred to another. That is generalized transduction. It may be a complete generalized transduction or abortive generalized transduction, that I told you in detail in the last class. Now we are going to discuss today about specialized or restricted transduction. Okay, see, specialized or restricted transduction is found mostly in temperate phages. Okay, it is extremely efficient as far as gene transfer from donor to recipient is concerned. Okay, and specialized transduction is found in temperate phages. What are temperate phages? Temperate phages are those which exist in prophage condition in their host. Now, what is a prophage? See, when a bacteriophage infects its host, then it releases its DNA inside the host. That means nucleic acid inside the host. When the viral genome gets integrated inside the chromosomal genome of the bacteria, okay, viral DNA or viral genome gets integrated inside bacterial DNA in that case and exists as prophage. In that case, the viral genome is called as prophage. That means DNA of virus or nucleic acid of virus integrated inside chromosome of bacterial host. Okay, such phages which exist in prophage conditions are called as temperate phages, and temperate phages show specialized transduction. Okay, now in some occasions DNA from a specific region of the host chromosome is integrated directly into the virus genome, usually replacing some viral genes. The resulting phages are defective temperate phages because they have bacterial DNA as a part of their genome. I'll tell you this in detail. When a bacterial cell is lysogenized by lambda phage, okay, we are going to discuss specialized transduction by taking an example of lambda phage of E. coli. Okay. Now, lambda phage of E. coli is a lysogenic phage. See, whenever bacteriophages infect their host, they either enter into lytic cycle or they enter into lysogenic cycle. So, lambda phage is a temperate phage of E. coli. It exists in lysogenic stage. That means lambda phage attaches itself to the host E. coli. Lambda phage releases its genome or releases its DNA inside E. coli. But lambda phage immediately is not capable of undergoing lytic cycle. So the lambda DNA gets integrated inside chromosomal DNA of E. coli and exists as prophage. That means it enters into lysogenic cycle and the lambda DNA multiplies along with E. coli DNA and is transferred to the daughter cells as if it is a part of E. coli chromosome. Okay, this is an example of lysogeny. This is what happens in lysogeny. That means viral DNA integrated inside bacterial DNA and it multiplies along with bacterial DNA. Okay. When the lysate containing, okay. Now viral DNA replicates under host control. Sometimes upon induction, okay, maybe exposure to certain kind of chemicals, maybe due to exposure to UV radiations or any such mutagens, that is upon induction, the viral DNA separates itself from the host DNA. That we say that initially the viral DNA integrates inside the host bacterial DNA. Now, because of induction, the viral DNA separates itself. The process of separation is called as excision, breaking out, cutting out. Okay, so it excisates itself. If the excision or separation is normal, then lambda DNA is separated and the host bacterial DNA is separated. But sometimes the excision or separation process is incorrect or improper. Okay, when the excision, lambda phage excision is incorrect or improper, the lambda phage, while getting separated from the host chromosome, will carry some bacterial genes with it. Okay, what I'm saying, when the excision of lambda phage, when the excision of bacteriophage from the chromosomal DNA occurs incorrectly or improperly, then the lambda phage DNA will carry few genes from bacterial chromosome. Such a lambda phage is a defective phage. Okay, the lambda phage DNA is, why it is called as defective? Because it is carrying few genes from bacterial chromosomes, right? Such defective phages are, rather we write them as lambda D. These defective phages are actually transducing phages. They are capable of bringing about specialized transduction. Okay, now why specialized transduction? Right? Whenever the lambda DNA attaches itself to its host, integrates its DNA, then the lambda DNA gets integrated inside chromosomal DNA of E. coli at one particular site. There is a site called as attachment site on lambda DNA. There is also an attachment site on bacterial DNA, that is E. coli DNA. With the help of the attachment site of lambda DNA, it attaches itself with E. coli. And the lambda DNA or bacteriophage DNA gets integrated inside E. coli DNA between galactose gene and biotin gene. Gal and bio. Between gal gene and bio gene. Gal gene means galactose gene and bio gene means biotin gene. So every time when the lambda DNA, bacteriophage DNA, integrates inside bacterial DNA, it will integrate between galactose gene and biotin gene. Now, whenever there is a incorrect excision, whenever lambda DNA comes out incorrectly, it will always carry either biotin gene or galactose gene. That means the defective lambda phage will carry only the adjacent genes, right? So, because say for example, if the lambda DNA has come out in an improper way and carrying the galactose gene, then it is called as lambda DG, lambda phages defective for galactose locus, because it is carrying galactose gene from E. coli. So specialized means, why specialized transduction? Because every time if defective phages are formed, they will have either galactose gene or biotin gene. Okay, so they can transfer galactose gene from donor bacteria to recipient bacteria and no other gene. So the name specialized transduction. So E. coli phage lambda is a classic example of specialized transduction that integrates its DNA precisely between operons encoding enzymes responsible for galactose and biotin utilization. So it integrates itself between galactose gene and biotin gene. Now let us discuss specialized transduction. In specialized transduction, a particular phage can transfer only a few restricted genes to the recipient cell, in contrast to generalized transduction. In generalized transduction, any gene from the donor is transferred by phage to the recipient. In specialized transduction, few restricted genes are carried by phages from donor to the recipient cell. This process was studied by Lederberg and Morse, and they found that in the lambda phage, the transducing activity was restricted to galactose locus. Only the genes adjacent to the insertion site are transferred. Other genes are not transferred. Okay. When a prophage, again understand that lambda phage DNA integrated inside E. coli DNA. In that case, it will be called as prophage. Okay. And this is shown generally by temperate phages. When the prophage is induced to leave the host chromosome, we induce maybe purposely, deliberately by exposing to UV light, or maybe naturally. When there is induction to the prophage, it will leave the host chromosome. It will excise itself from the host chromosome. And sometimes the excision is improper. The resulting phage genome contains portion of bacterial chromosome. About 5 to 10% of the bacterial DNA next to the integration site. Okay. So by mistake, because the excision is improper, it will carry 5 to 10% of bacterial DNA. But which DNA? Adjacent to the lambda DNA, and most of the time it is galactose gene. A transducing phage genome usually is defective. It lacks some part of its attachment site. The transducing particle will inject bacterial genes into another cell, that is recipient cell, through the defective, though the defective phage cannot reproduce, but the defective phage can transfer DNA or bacterial chromosome from one bacteria to another. Okay, the best studied example is, as I told you, lambda phage. The lambda genome inserts itself into the host chromosome at specific location known as attachment or at site. Okay, the phage at site and bacterial at sites are similar and can complex with each other. As you can see in this diagram, this is lambda phage shown in red color. There is attachment site, okay, and this of lambda phage, and this is the attachment site of E. coli. Now with the help of the attachment site, there is crossing over, right? And in the next diagram, you can see that the lambda DNA is integrated inside E. coli DNA. Okay, the red colored lambda DNA is integrated inside E. coli chromosome, and you can see from the diagram that the lambda DNA is integrated between galactose gene and biotin gene. So this is lambda DNA, okay, integrated between galactose gene and biotin gene. Now when the lambda DNA separates, when this lambda DNA separates properly, it will be removed or excised because of induction. But if there is a mistake in excision, the lambda DNA will carry with it the galactose gene. Okay, leaving a part of lambda DNA into E. coli chromosome, and it will carry the galactose gene. Why it will carry? Why we call it as only few genes are transferred? Because genes adjacent to the lambda DNA are only taken up by the lambda DNA by mistake. So in normal looping out, phase is excised out. If there is rare or abnormal outlooping, lambda D gal, that is lambda phages defective in galactose gene, are produced. Okay. Now you can see from this diagram, the first one, there is normal looping. Look at the first normal looping, okay, where the lambda DNA has separated itself from the chromosomal DNA of the host. You can see that this is normal looping. So the lambda DNA is separated properly from the host DNA. So host DNA is separate, lambda DNA is separate. When this normal excision takes place, defective phages are not formed, and transducing phages are not formed. The phages cannot carry out transduction. Now in the second diagram, when there is rare or abnormal looping, what will happen? You can see that there is the excision or separation of lambda DNA from the host is defective. And you can see in this diagram, second part, that lambda phages have carried with it, in the second part, lambda phages have carried with it the galactose gene. Okay. So lambda phages are having galactose gene from E. coli. That's why such phages are called as lambda D gal. That means lambda phages defective for galactose gene. Lambda phage genome, bacteriophage genome is having chromosomal genes of bacteria. Okay, so virus having bacterial DNA, such phages are called as defective phages, and these defective phages are capable of bringing about transduction. Right, once again look at the various steps of specialized transduction. You can see that there is a temperate phage which has released its DNA inside the host. The temperate phage genome gets integrated inside chromosomal genome, chromosomal DNA of the host, that is E. coli. In this case, it is called as prophage. Here it is called as prophage. Okay. So lambda phages existing as prophage. Now what happens? Because of induction, the prophage gets excised. You can see that. See in this diagram, the prophage has excised itself from the chromosomal DNA of the host. But the prophage here is having one part of chromosomal DNA from the host. You can see the prophage is having, the lambda DNA has separated out, but it has picked up a part of bacterial gene. Right, and hence the bacteriophages that are formed are all defective phages. You can see in every bacteriophage particle, it is having some part of the host genome. Such phages are called as defective phages or lambda DG gal if they are having galactose gene. Now, when this defective phage infects a recipient cell, it injects its DNA inside the host, and the DNA from the virus gets integrated inside the host. When the DNA from the phage gets integrated inside the host cell is having donor bacterial DNA. The host cell is having donor bacterial DNA. You can see here, right? So, the efficiency of transduction taking place or recombination taking place by specialized transduction are very high, right? So, the sixth diagram shows that bacteriophage genome carrying donor bacterial DNA inserts inside recipient genome, and the recipient cell undergoes transduction. That means it is having genes from donor, chromosomal genes from donor DNA. You can see here. Is that clear? So this is about specialized transduction. So in specialized transduction, the phage undergoes lysogeny usually at specific location, that is attachment site, and if the excision or separation of the prophage from chromosomal bacterial DNA is defective, it will produce lambda D gal. The recipient cell expresses itself. Now, there are two types of transduction: low frequency transduction and high frequency transduction. Right. Because the number of defective phage particles, that is lambda D gal, produced in a cell lysate are very, very low. That is 1 in 10 raised to 5 or 10 raised to 6, or rather we can say 10 raised to minus 5 to 10 raised to minus 6. The frequency of formation of defective phages under normal condition, under natural induction process, is very low. And because defective phages are formed in very low number, the frequency of transduction is also very low. Such a lysate is called as low frequency transduction lysate. Now, lambda D gal has the capacity to convert gal minus cells into gal plus cells. But the frequency is low because the number of defective phages formed are less under natural natural induction process. So such a type of transduction is called as low frequency transduction. But if we want that transduction to take place, we can increase the frequency of transduction. Okay, so those lysates in which there are a high number of defective phages is called as high frequency transduction lysate. How can we do that? By exposure of the lambda phages to integrated lambda phage to UV light. Okay, now let us understand here the lambda phage genome shown in red color integrated inside between the galactose and biotin gene of E. coli. Now because of induction, the lambda phage genome got separated. So here in this diagram you can see normal lambda phage genome separated from the E. coli chromosome. In the second part you can see, so here we can see normal lambda phage separated, and in the second diagram you can see lambda phage has separated but it has carried with it galactose gene. Okay, lambda phage, this is called as lambda D lambda phage defective for galactose gene because the lambda phage has excised or separated from the host chromosome, but it has carried what? Galactose gene with it. Such a phage is capable of carrying out transduction, but the frequency at which the defective phages are formed is very low, and hence the chances of transduction taking place are less, so they are called as low frequency transduction lysates. But we can increase the frequency. So now high frequency transduction, you have to expose the lysate to UV light. So under controlled condition we can expose it to UV light. Okay, and the number of defective phages formed will be very high and hence have high transducing power. Okay, nearly half of the lambda phage particles, 50% of the phage particles have the capacity to transduce gal minus cells into gal plus cells. If your cell is deficient in beta galactosidase gene, and if it is infected with lambda D gal, the gal minus cell will be converted into gal plus cell. The term high frequency transduction HFT is used for such lysates which have the ability to bring about transduction in high number. Okay, HFT lysate can transduce only gal gene. It has the ability to transfer only gal gene from donor to the recipient. Now, if there is a normal lambda phage genome in the same cell, okay, if the recipient cell is already infected with lambda phage but normal lambda phage, still the defective lambda phage carrying the gal gene can integrate. Okay, so the cell is already having lambda DNA integrated inside its chromosome, but still the lambda defective phage can get integrated, and such a cell having 50% of the you can say chances that it is called as high frequency transducing phage because the frequency is very high because it is having a supporter lambda phage as well as the defective lambda phage. Okay, so it can bring about transduction at a very high frequency, and hence it is called as high frequency transducing lysate. Okay, so that's all with transduction, and hope you understand. If you have any problem, hope you understood, and if you have any problem you can call me.